Hi, welcome back to the Anand Medical Academy online biology sessions. Let me continue the molecular base of inheritance after transcription. After transcription, result of transcription, there was a matured mRNA called mRNA. Where does transcription takes place? Inside the nucleus in case of eukaryote. Whereas in case of prokaryote, it occurs in cytoplasm. But let me discuss the eukaryotic translation process. So that transcription occurs inside the nucleus, then through nucleopores it comes out. Nothing but mRNA is migrating from nucleoplasm to cytoplasm. Once mRNA arrives to the cytoplasm, then protein synthesis starts. The protein synthesis is called translation process. During translation, what is happening? RNA is directing the synthesis of polypeptide chain. Polypeptide chain means what? Protein. Many amino acids are linked by peptide bonds together form a polypeptide chain is called protein. Let us see how this process runs. Here I would like to tell you, in this process all the three types of RNAs are involved. mRNA, tRNA and RNA. But mRNA acts as template for protein synthesis and tRNA acts as cover leaf, is also called adapter, who helps to bring the amino acid, and RNA helps to catalyze. So it has many catalytic activities. You know very well about all the three structures. I will give you roughly. This is the mRNA, which is a translator from template strand of DNA. The tRNA is look like cover leaf structure. It has many arms. And this is the 3 prime, this is the 5 prime. Here OH will be there, here phosphate will be there. It's a cover leaf structure that belongs to tRNA. This is mRNA. Whereas if you see RNA, it has 2 subunits, one smaller subunit as well as one larger subunit is present. During protein synthesis only these two will be assembled, otherwise they will be individual. Clear? And this process will be explained very easily by 3 manners. One is initiation, means the starting process of the protein synthesis. Later elongation, how long the polypeptide chain is forming, that is called chain of elongation. Lastly, how this chain is going to be terminated and decides the length of the protein is called termination. You have to understand all the 3 things. You have to know very basic concepts. Let me discuss the basic concepts now. The first step to understand all the process is activation of amino acids. Generally, when we eat food, in that proteins are present. Such proteins will be digested in stomach as well as intestine. You know, you have learned that in the process of digestion and absorption lesson in your first year. They get absorbed into the blood through the liver. Then these absorbed things are not proteins. Proteins will be digested and result in amino acids. Such amino acids through blood reaches our tissue, nothing but cells. Inside the cells, amino acids are existing inside the cytoplasm. In the cytoplasm, the existing amino acids are inactive. To active the amino acids, such a process is called activation of amino acid. I hope you understand. Clear? Even what I am going to convey is, even we took a protein by the food, that amino acids not directly assemble the protein. The process is required. That is what I am trying to explain. Activation of amino acid means generally an amino acid, anything it can be. Suppose methionine. Why? Because it is the very first amino acid. Methionine is an amino acid. This methionine or any amino acid required one ATP, nothing but adenosine triphosphate. To active this amino acid into activated form, what is required? Enzyme is required. That enzyme name is acyl synthetase. Acyl synthetase. Acyl synthetase is a enzyme. That's nothing but amino acid synthesis. Later we will come to it. So one amino acid plus one ATP plus one enzyme gives activated amino acid, that is called amino acid plus amino acid means what? Activated amino acid plus AMP is how all amino acids are activating. You know very well the very first code of mRNA should be AUG. Why? It is called starting codon. So AUG declares which amino acid? Methionine. So that if the methionine is activated by the same process, what is that? That enzyme is called acyl synthetase. Then methionine will become formyl methionine. In short form, it is called FMET. For any protein synthesis, the very first amino acid is methionine. For the process of activation of methionine is called formyl methionine. In what is the very first amino acid to initiate the process of translation. Let us see, once amino acids are activated, what is going to be? Once amino acid is activated, that will go and bind to tRNA. Where exactly it binds to tRNA? Then how this process runs? Let us see. You know very well the tRNA structure, covalent structure. It has many arms. This is the 5' and this is the 3'. At 3' OH will be there, so that here only amino acid will come and bind, that we are seeing in the structure of tRNA. Right. So activated amino acid formula what? A means amino acid plus AMP plus enzyme. This is what called activated amino acid. Is also called amino acyl complex. Generally, to activate one amino acid, 1 ATP is required. After activation, AMP is coming. ATP means how many phosphates? 3 phosphates. Adenosine, correct. Activated amino acid consists of what? AMP. AMP means what? Adenosine monophosphate. So ATP minus AMP, 3 minus 1 is equal to 2. So 2 phosphates will come out. But activated amino acid is also called amino acid complex. It is nothing but one amino acid and AMP plus enzyme. This one is going to bind with the clover leaf structure 3'. Why? Their functional group is called OH. So tRNA brings the amino acid in the process of translation. Means it has to receive first. Clear? So amino acid is going to bind at OH and R3' of the tRNA. That equation let us see. AA plus AMP plus enzyme all together is called amino acyl complex plus tRNA gives rise to amino acid having tRNA. So this is what called binding or transfer of amino acid to the tRNA. This is the second important step. The first step is in cytoplasm, amino acid should be activated, followed by transfer of activated amino acid, not normal amino acid. Normal amino acids cannot attach to tRNA. Remember, once amino acid is activated by the process I just told you, then that activated amino acid only will go and bind at OH end or 3' of the tRNA. Then the second process is completed. Next, what will happen? Let us see. In the third step, protein synthesis takes place by another 3 interfaces. Assembly of RNA followed by attachment of mRNA and finally polypeptide chain synthesis. Let us see all the 3 now. Assembly of RNA. Generally you have seen RNA as a small unit and one large unit. In case of eukaryotic, that is an 8S ribosome. In case of prokaryotic, that is 7S ribosome. So let us see the first step here. This is the smaller ribosome unit and this is the largest ribosomal unit. This is a larger unit. This is the smaller unit or a small unit. Okay. In case of eukaryotic, it is a 60S and 40S. In case of prokaryotic, it is a 30S and 50S. Okay. Now, in the larger subunit, there are 3 sites are there: A, P, and P. A means amino acyl site. P means peptide site. E means exit site. Very important. If you not understand all the 3, in final step of protein synthesis, you will be confused. Repeat again. The first one is assembly of RNA. Protein synthesis need assembly of RNA. RNA is not a single unit. It smaller subunit and one is larger subunit. So larger subunit, inside there are three regions are present. For our convenience, we can call up them A site, B site and E site. What is A site? It is amino acyl site. Second one is P site, means peptidyl site, which helps to form the peptide bond. And what is E site? Once a peptide bond is formed, the E site helps to exit the tRNA that comes out of both the complexes. Clear? In the process you will understand. And secondly, attachment of mRNA. And where does the mRNA is going to attach rRNA? But rRNA has two units. One is larger subunit and another one is smaller subunit. To which unit mRNA is going to bind is a very important thing. Answer is mRNA is binding to smaller unit. So smaller unit have the binding site of mRNA. On that, this mRNA will come and bind. So this is what mRNA. mRNA is attached to which subunit of the tRNA? Smaller subunit of the tRNA helps to bind the mRNA. That's a clear. Next one is polypeptide chain synthesis. Polypeptide chain synthesis means what? Let us see the formation of these two means assembly of the RNA required magnesium ions. So magnesium, the presence of magnesium ions, only the smaller and larger subunits are going to be assembled. Once these two are assembled, mRNA will go and bind to the smaller subunit of tRNA. Generally on mRNA codes will be present AUG, GUG, CCC, so many will be there. According to the codes, anticodons are present at the tRNA. This is the anticodon loop. So that already before I told you that the tRNA helps to bring the amino acid. Once amino acid is attached here, according to the anticodons, the tRNA will come and bind at the mRNA codes. So that anticodes present at the tRNA are opposite to mRNA codes. Let us see how this polypeptide chain synthesis will form in detail. See guys, everyone should know that the very first codon of the mRNA is AUG. Why it is called starting codon. So, but AUG is read by P site. Very important. Except AUG, all codes are read by site A. This is a very important thing. Means only AUG is read by site A. Other than AUG, all are reading by site A. Means first tRNA is coming and bind to the mRNA opposite to T. Later everything is coming and bind and opposite to A. After some time I will provide a visual in that you will understand very neatly, no need to confuse. Okay, once mRNA is attached to the smaller subunit of RNA, polypeptide chain synthesis will be started. Suppose if this is the mRNA, then this is the small subunit and this is the larger subunit. This is the site P and this is the site A and this is the site E. The first tRNA comes along with one activated amino acid and binds at site of P. Why? Opposite to P, AUG will be present. Next codon is present. Second codon of mRNA is present. Opposite to A, that will be UUU. Understand? So, suppose this is one tRNA. Who is bringing very first amino acid? That is nothing but methionine. Then this tRNA is called T-met. Means tRNA which is bringing methionine or forming methionine F-met. This will have the anticodons that are opposite to AUG, means UAC. A opposite U, U opposite A, G opposite C. UAC anticodon, UAC anticodon having tRNA can bring methionine. Understand? Anticodons present on the tRNA opposite to the sequences or genetic codes present in the mRNA. So here the particular tRNA will come here and bind like this. Then this is A opposite U opposite A, and G opposite. See, all the three are compatible and just like an adapter. That's why the tRNA molecule is also called adapter. This tRNA molecule bind with the mRNA sequences where the exact amino acid is present. Here amino acid is present. What is that? Methionine. Next, second tRNA brings another amino acid according to the sequences of the genetic, first genetic code or starting codon. So U opposite, what will be there? If you take another sequence of anticodon of another tRNA, U opposite, what will be there? A will be there. A will be there means here phenylalanine will be present. Means opposite to U. So here another amino acid is coming. Second code is reading by side A. Then this is the another tRNA who brought phenylalanine. So once the two tRNAs are added side by side, then by the help of side P, peptide bond will form between two adjacent amino acids. Once peptide bond is formed, then this first tRNA who brought the amino acid will be released out. By the site of E. That's why site E is called exit site. It helps to release the tRNA once the peptide bond is formed. Here, what is the function of P site? Formation of peptide bond. What is the function of A site? Receiving of tRNA. What is the function of E site? Exiting the tRNA after formation of peptide bond. So now, what will happen? Generally, students will think that the mRNA is sliding. No. On the mRNA, both the smaller and larger subunits are riding. Means nothing but they are sliding. So it comes here and it comes here. Then this is the P side, this is the A side, and this is the E side. So next tRNA component, activated amino acid along with that, it will come and add to A. According to the sequence of something C, then there will be G, then some amino acid will be attached at its 3' end, then it will come and bind here, just like that. Here another amino acid is present. Once it is attached to the codon, here peptide bond will be formed by the side P, then this tRNA will be released out. Like that, after formation of every peptide bond, one tRNA will be released from the site E. Peptide bond is formed by the help of site P. tRNAs come and bind at the site A. This is how A, P, E are helping. A helps to receive the active amino acid now. That's why it is called amino acids, right? P helps to form the peptide bond between amino acids now. That's why it is called peptide site. E helps to release the tRNA once peptide bond is formed. That's why it is called releasing site or exit site. So, like that, both the subunits of RNA are riding on one direction of both the mRNA sequences, while tRNA is coming and releasing through the larger subunit. That time, a lengthy polypeptide chain is forming. My question now is, how long this length is formed? Unless a stopping codon. What are the stopping codons? Are 3. Out of the UAA, UGA, UGG, these codons, if any of the 3 comes, then there is no translation process. So that are called untranslated regions. They are called UTR. Once untranslated region comes, then there is no another tRNA comes and attaches to the mRNA. Whenever there is no tRNA comes, there is no peptide bond formation. Then ultimately the chain of polypeptide or the chain of protein will be decided. So from starting codon to stopping codon, how long the mRNA expresses its codons along with the help of tRNA as well as both the units of RNA, polypeptide chain will be released. Regarding this concept, I am going to provide a visual now. You just see and understand the concept. In this video, you have to understand once the proteins are digested in our body, how they becoming amino acids. Even such amino acids are reaching our cells by blood. Then there are inactive amino acids. How these inactive amino acids are becoming protein by the process of Translation, let us see.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
So guys, by this visual, I hope you understand everything about the translation. Listen the class once again and also refer the NCERT textbook and attend the assignment. Thank you.